Welcome to our third annual research symposium, and thank you from the Society of Conservation Biology for coming out today. And our opening speaker this morning is Dr. Fred Allendorf. And Fred has been a professor at the University of Montana since 1976. And since 2004 has received the honor of being designated a Regents Professor at the University of Montana. He received his bachelor's in zoology at Penn State, MS in fisheries at the University of Washington, and PhD at the University of Washington as well. He has published extensively in the Journal for Our Society, which is called Conservation Biology, and in fact, he did a quick Google search, and since 1988 he has published 21 papers in the journal. So we're really lucky to have him here this morning, and with that I'd like to introduce Fred Allendorf.
Thank you, Jody. Can I say one thing? Yeah. Fred thinks the Phillies are going to win the pennant.
Of course, they are. I didn't think so yesterday, but I do today. So I didn't make this title up. Somebody else made it for me. And Jody asked me a couple days ago, are you going to say it all started in Philadelphia? And I said, it depends how the Phillies do the next couple of days. So I will admit I'm from Philadelphia. So I didn't make this title up, but you can tell you're really old when somebody else makes up a title like that for you. And like Jody said, I've been here since 1976, and I also have a partial appointment at Victoria University in Wellington in New Zealand. So what I'd like to do today, just a little bit of introduction, and then talk about, because I really was there at the beginning of conservation biology. So Joel, did you go to the first conservation biology meeting in Bozeman in 86 or 87? Why not? I
wasn't part of the clique.
Oh, okay. Well, the first meeting actually was joint with evolution. And that was really, at that point, the main reason I went. It was joint with the evolution meetings, but it was hosted in Bozeman by Peter Broussard, who then became Joel's chair, or vice versa. So I was back at the beginning of conservation biology, talk a little bit about that. So the only thing I know about other than baseball is genetics. So I'll be talking primarily about genetics. And I think the most valuable part of my knowing about baseball is when my wife does crossword puzzles. I can always answer. Joel's pretty good too, though. He's amazing. So then I want to talk about the history of approaches to detect genetic variation, because that's been a big part of trying to use genetics to understand natural populations and conservation. And then two case studies where some of the work done by people in my lab have been applied to conservation problems. One in which we've been successful in getting the science incorporated, and one where we have not been successful in getting the science incorporated. So geneticists and myself were fascinated by variability. So when I sit in a room like this, I often look at people. You see men and women, different color hair, different ethnic backgrounds. So genetics is really the study of variation within populations. And most biologists, I think, often see variation as a problem. So if you're writing a field guide to birds, it's really a pain when you have to put multiple pictures of birds that have different coloration. Or if you're writing an anatomy book, having to write down what the differences are between individuals can be difficult. So a lot of biologists would rather there weren't variation, but genetics is based on variation, and most species have quite a bit of variation. And there's one of my favorite pictures of variation. Wilt Chamberlain, who grew up not far from where I grew up in Philadelphia, and Willie Shoemaker. So, and this is a nice picture because it shows both, genetic variation within species, but also shows genetic variation between different racial groups or different populations from different parts of the world. Of course, today what geneticists are really obsessed by is not looking at things like that, but looking at things like A, T, G, and C, looking at DNA sequences. And back in the beginning, when people began to apply genetics to problems in conservation, it began as captive breeding. So almost all the people working on genetics in the beginning of conservation biology was something like this with tigers, bringing tigers into captivity. But I guess I've never been a big fan of captive breeding, and I think conservation genetics has become a much healthier discipline as it moved to try to deal with trying to conserve species in the wild and natural populations. And so this was an effort to try to conserve tigers in the wild, 2005, 2015. As far as I can tell, talking to my friends who know a lot about tigers, it hasn't been overly successful. But at least I think the effort is in the right place, trying to preserve animals and plants in the wild rather than in captivity. So my interest is, I'm primarily a population geneticist, evolutionary geneticist, but I'm really interested in applying genetics to problems in conservation. And this is a book that Gordon Lukart, one of my former students, and I wrote a few years ago. And it really is, from my view, it's a population genetics book using examples from issues in conservation. So this is the early days of my career anyhow. So I did my PhD, as Jody said, at the University of Washington, and it was a joint degree in fisheries and genetics. And this was before conservation biology, before there's anything called conservation biology. But I always had, it was sort of serendipity that I did, because I went to the University of Washington. The person I was going to work with had a project on rainbow trout. I was interested in evolution of polyploidy in salmonids and rainbow trout. But my funding came from Washington Department of Game. And what I did was primarily look at genetic differences among populations of steelhead in the state of Washington. So the paper, the gene duplication paper, is published in Genetics, a basic genetic journal, but at the same time as I was also doing applied work and trying to apply this to issues in conservation. So this has been part of my whole career, and I really feel that genetics, I feel fortunate in population genetics to be able to do this, to really deal with the interesting scientific, basic scientific issues, but also have them be very important for applied and issues that we're concerned with as a society. And the beginning of conservation biology, really I think most people would say the conservation biology began with Michael Soule, here on the right, Scott Mills, major professor. And conservation biology was really founded with a series, probably the two books on the left, that were edited volumes by Michael Soule and other people. And I think that's where the name conservation biology first came from. So he was a Paul Ehrlich student, worked in San Diego, and became aware that most of his field sites were quickly disappearing, and became concerned about those issues, and then tried to get basic, well, it did get many basic ecologists and geneticists interested in problems in conservation. And this is to remind me that in the early 1980s, when I was here in the zoology department, I tried to introduce a course called conservation biology. And I remember the faculty meeting very clearly that Lee Metzger, some of you may know Lee, was a faculty member then. And Lee has done a lot of good work in conservation biology. He was involved in issues with the Yellowstone elk and bears and other things. But when my course came up, Lee said, there's no such thing as conservation biology. You can't teach a course with that name. And I never really did understand what Lee meant. So I gave him a call yesterday and I talked with Lee, and he denies ever saying it. But he did, I'm sure he did. And I think at that time, some people, I think, didn't like the idea of conservation biology because they felt that's what wildlife biologists had been doing forever. And I'm not sure that's what Lee felt, but I know that for some reason he did not think that conservation biology was a real biological discipline and didn't want me to teach a course called conservation biology. So I like to go back to the beginnings, and the beginnings of conservation genetics was Darwin. So Darwin wrote a book that probably most of you here haven't read, even if you've read The Origin of Species. So Darwin was interested in plants and animals under domestication, and most of what he knew about genetics came from practical breeders, either plant or animal breeders. And this was a 2-volume collection, it was a tome. And chapter 17 was entitled On the Good Effects of Crossing and the Evil Effects of Close Interbreeding. So there's two things here. You know, we don't write scientific papers today talking about the evils of inbreeding. And the other is that when he said interbreeding, today is what we would call inbreeding. So the word has almost completely changed its meaning. And the reason that I think he is the first conservation geneticist is because he thought about applying genetics to issues in habitat fragmentation. So fallow deer were only found in hunting parks around England, and he realized they had small population sizes. He realized that small population sizes meant matings between relatives. He realized that matings between relatives often produced progeny that had reduced survival and other problems. So he went around and talked to the managers and found out that the managers, even in the 1800s in England, were moving males around trying to get out of these issues. And this is sort of a preview of what I'm going to be calling genetic rescue later. And the first real paper in conservation genetics was Sir Otto Frankel, who was an Austrian who moved to Australia, and he was a plant geneticist interested in agricultural crops. He wrote a paper published in 1970, and it was his interest was primarily agricultural crops, but he also was aware that these same principles and issues applied to wildlife. And I really like this table, the time scale of concern. For the genetical conservationist, the time scale is 10,000 years plus. For the politician, it's the next election. So things haven't changed very much in 40 years. And this is a paper that Gordon and I and Paul Hohenlohe just published, sort of bringing conservation genetics into the genomics era. What can we do now with a lot of these new techniques in terms of applying them to issues in conservation? So that's a 5-minute overview of conservation genetics. And you can it's really easy to make lists like these. What are some of the issues that conservation genetics deals with? So number 1 is captive populations. That's really where conservation genetics began. Description of individuals, relationships among populations, taxonomy, looking at the effects of habitat fragmentation, looking at the effects of hybridization, looking at local adaptation, the importance of local adaptation from a conservation perspective, just using genetics to sort out the basic biology, even if you're not really interested in genetics, it's just a valuable genetics tool, forensics, and finally, genetic engineering, which I'm not a big fan of in any sense, but I really think there are some cases in trees, long-lived trees that are infected by diseases. The generation interval in a tree is so long that it's really hard for a tree to evolve resistance to diseases. And plants often have a simple one gene, one disease relationship, and so I think theoretically, you could imagine where genetic engineering resistance to diseases could be valuable for conservation in trees. Today I'm going to talk about the grizzly bears, looking at the loss of habitat loss fragmentation, and then I'm going to talk about Westlope cutthroat trout, where the issue is hybridization. And I'll also talk a little bit about forensics, just using genetic techniques to identify species, populations, or individuals, and how that can be used in forensics. So genetics really began when Mendel's principles were rediscovered in 1900, but for 70 years, there really wasn't any way to go out and look at genetic variation out in natural populations. So it was laboratory matings and chromosomes. So people used Drosophila, fruit flies, sometimes mice, but really mice was not a wild population. The only natural genetics of an animal was Drosophila until 1970, when biochemical techniques came along and have completely revolutionized our understanding of genetic variation. And the first technique was protein electrophoresis, or allozymes. And again, looking at the history of conservation biology. So allozymes used to be called find 'em and grind 'em. People just went out, collected tissues from any individual and ran them. I'll show you briefly how the techniques worked. And so it really was, from the beginning, which was 1969, a flood of papers as people just went out and collected more and more individuals, more and more species. And I began my graduate career in 1971, so I really was an allozyme baby. So at the beginning of my career, everybody interested in population genetics was beginning to use protein electrophoresis. So for the first time, we could look. We didn't have to study Drosophila if we wanted to be a population geneticist. And so here is the technique from a fish perspective. I worked in a fish lab. So you could go out and collect lots of individuals. One of the troubles with allozymes is that it was destructive sampling. So you had to look at different tissues. So you would take a piece of muscle, a piece of liver, fluid from the eye, because you were looking at proteins, and different proteins are expressed in different tissues. And then you could go through a fairly simple lab procedure, so simple that even I could do it. And then you could end up with these dots on a gel which represented genetic differences between individuals. And this is one for Chinook salmon. So there are 12 individuals here. This is one particular enzyme that people looked at using protein electrophoresis. And you can see that, so individual number 1 has a single band up there, so that individual was homozygous for a particular enzyme allele. Individual 2 has 2 bands. Individual 2 is heterozygous. Individual 3 has, again, a single band, but it's the lower band. So the rate that the proteins are moving through this gel is a function of their charge. Enzymes that have different charges are genetically different. So you can look at genetic variation in a lot of individuals at a lot of loci fairly quickly. And the nice thing was you could use this exact same techniques pretty much in any species. And so there was that huge boom of papers that began in the 1970s. And this is just one example of a forensic example of using allozymes. So this was work done by Jim and Lisa Sieb. Jim was a fellow grad student of mine. Lisa was a student of mine when she was here, got her master's degree looking at king crab in Alaska. So there was a poaching issue. So there were different areas of king crab in Alaska, and the fishery down here was open, so people were allowed to catch crabs down here, but a boat came into dock, and Alaska Department of Fish and Game suspected that they had caught those crabs in an area that was closed up in the Bering Sea. So just looking at them morphologically, you can't tell. They went in, took, 100 individuals, took proteins or took tissue samples from them, ran them in the lab, and Jim and Lisa had baseline data. So these are 5 different loci. Just the amount of coloration represents genetic differences. So there are genetic differences between Southeast Alaska and the Bering Sea. So for example, looking at this first locus, ALP, the dark allele is about 80% or more, where it's only 20% up in the Bering Sea. And if you look carefully, you can see that the frequencies that they found on the boat were identical to the ones from the Bering Sea. And what happened was this guy pleaded guilty because he knew he was caught, and he paid a fine of, I think it was about $75,000 or $100,000. And at that point, that was enough to start a genetics lab. And so that's what ADF&G used to start their genetics lab. So now, we don't have to go out and grab tissue samples. Everybody, I think, knows about DNA, or polymerase chain reaction. So this was a picture I took, not on that bus, but a very similar bus when I was in Perth, where spitting is unacceptable. Drivers are now equipped with DNA kits to help with the apprehension of offenders. So I never saw a driver, if I should have went up and at in front of the driver to see, but somehow the threat that if you spit on my bus, I'm going to look at that DNA and we're going to nail you. But and my point here is that we now can get genotypes on virtually anything which has a few cells in it. So there's a paper published. So things like whales, you can now do genetics of whales by the skin they slough off when they dive. There's a paper published about a month or two ago. They looked at dolphins in captivity, and they collected the breath, and there's enough cells in the breath that they could actually genotype individuals. And this is because of the polymerase chain reaction, which I would guess most people in here have certainly heard of and know a lot about it. So polymerase chain reaction is just a way that you get one copy of DNA by going through thermal cycles, heating and cooling, using the right enzymes, Taq polymerase. We can start with one copy of the DNA and produce billions of copies by going through this process of doubling. And I think probably most people here know that the enzyme, the key to this enzyme is that it functions at very high temperatures, 60, 65 degrees C. It doesn't break down. And so that enzyme was found in microbes living in hot pools in Yellowstone. So a similar approach to what done with Alaskan king crabs, more sophisticated, looking at whales. This was worked on a while ago by Scott Baker and Steve Palumbi. So people suspected, so the Japanese, I think as most people know, are allowed to do research harvest of southern minke whales, and then they're allowed to sell that meat. But people suspected that they were selling other kinds of whale meat as well as southern minke. And so this is Matt Hare. He was a postdoc at that point. They couldn't analyze, so analyzing this meat genetically, even using allozymes, would have worked. But if any genetics lab in Japan had done the work, they would never have gotten any money again from the government. It was legally, because of CITES and other agreements, they couldn't take the samples of the whales out of Japan. So what they did was they went to the fish market, grabbed samples, went to the hotel room, did PCR, took the copies of the DNA, like Xerox copies, and then they left the original whale meat in their hotel. And then they flew back to Hawaii and Virginia. New Zealand, up top shows this is an automatic sequencer. They did sequencing, and then just using a similarity cluster analysis, all these ones called samples are the unknowns that they got in the fish market. And if you look at how they cluster with known whale specimens, there were about half of them, almost half, were what they were supposed to be, southern minkies. There were a couple of unknown dolphins, another unknown whale. There were 4 fin whales, a humpback, and a northern minke. So, and this is still a big issue in trying to figure out what whale meat is being sold. And this is a simple forensic application where you're just using genetics to identify the species by looking at the DNA sequence. And this is sometimes called barcoding. You may have run into this. It's really not very interesting, but it's a neat story. I mean, it's genetically not very interesting. More interesting is looking at individuals. So here we have microsatellites. If you look at Jodi's poster out there, she talks about microsatellites. So each column is an individual, and you can see, so individuals either have one band for each locus, here are 3 loci, 3 genes, or 2. And if you look across there, I would guess that every individual has a unique pattern. So rather than just identify species, we now rather easily can identify individuals, and this has lots of uses. So if you work for the FBI, they use it a lot in criminal investigations. So when Monica Lewinsky kept her dress in her closet for several years, they went back and took the sperm samples and looked at the microsatellite pattern, and then they collected sperm samples from President Clinton, and they found an exact match. And the probability of two people at random in the population matching was 1 over 7.87 trillion. So this was fairly strong evidence that somehow Bill Clinton's sperm got on Monica Lewinsky's dress. And Jeff Stetz is talking at 20 after 10. He's going to be talking about using this same approach, trying to identify individual grizzly bears, collecting hair samples and other kind of samples. And this is a paper that Jeff and others published a year or two ago, estimating population size. And this is, I don't want to talk about what Jeff's talking about, but this is based upon being able to identify, you go out and collect a bunch of samples, you can identify how many unique individuals you've sampled, and by that number you can come up with estimates of how large the population is that you are sampling. So I don't really find that interesting either. I like biology, not, well, I like genetics. So those are valuable uses of genetics, but they don't really get at the underlying genetic principles. I think more interesting is what difference does genetics make in terms of whether populations are locally adapted, whether they're viable or not. And just a simple view, the extinction vortex was something coined by Michael Soule way back in the early 80s. So we have a large continuous population. We lose habitat, ends up becoming fragmented and isolated. So now we have a small population size. That leads to increased inbreeding and genetic drift. Darwin knew about the inbreeding. He didn't talk about genetic drift, but it's the same principle that Darwin was talking about with fallow deer. Darwin knew that inbreeding causes decreased reproduction and survival. And sometimes, not always, this could lead to reduced population growth rate, which in turn could lead to even smaller population size. So the idea, and this idea is overly simplistic and naive, but there's truth in it, that when populations become small, there is this feedback between the demography and the genetics. And those genetic concerns can increase the probability that populations are not going to persist for a long time. And this is one of my favorite bumper stickers that I first saw in Yellowstone Park, and I went to get my camera and came back and the car was gone, but then I saw it again up in the rattlesnake parking lot. And I really like this. Country music is not allowed in my lab. And we've also done a lot of work on developmental abnormalities associated with inbreeding. So this is really a great bumper sticker. So I thought maybe Sterling Miller would show up today. So I figured I had to do something to talk about why people should be concerned about genetics. So what's the evidence that genetics makes a difference? And it has been difficult. You can't go out and do the kinds of experiments we'd like to in wild populations. But I think the best evidence of that genetics sometimes, when populations are small, can be a real important issue is what's been called genetic rescue. And I'm just going to use one example of a snake in Sweden. And sort of the seminal paper on genetic rescue was written by these guys. And one of the, I think the most things I get most satisfaction from in my career are the students that have gone out in the world after working with me and Scott Mills. Dave Tolman worked both with Scott and myself, and Gordon Lukart was a student of mine. So the evidence, genetic rescue, so this is a paper published in Nature 10 years ago now, 11 years ago now, of a snake population which was on a small island. They knew the population size was very small. So this is 1981 down to 1992. The hatches are numbers of new recruits, and the solid bars are total numbers of males I captured. So you can see the number of males captured and the number of recruits was going down. Then they did a really nice experiment. They introduced males, and males demographically have no effect, but genetically, if inbreeding is a problem, males are going to have a big effect. So once they introduced males, potentially the inbreeding went away, and the inbreeding depression could go away, and the population quickly rebounded after that. So that's what we mean by genetic rescue. Here's a population that was in trouble going down the demographic vortex, and by introducing a few individual males, there was this recovery. However, we'd like to avoid the need for rescue. And so how do we know when if a population is going to be viable or not? And this is a computer program that's used to model population viability. And this is another good story. So the 76 National Forest Management Act said the Forest Service had to maintain viable populations of vertebrates. So I remember I got a phone call from Bob Shoemaker, who has worked for Fish, Wildlife, and Parks in Kalispell. He was a regional director, and he said, Fred, I have a problem. The Forest Service is interpreting viable to mean you need at least one male and one female. And he said, could you write a justification of why we need more than two individuals in a stream of cutthroat trout for it to be viable? And so I did, and I've been looking for that letter for years now, and I can't find it, but I know I wrote it. But it's interesting. I think we've come a long way from the perspective of thinking that one male and one female is enough for a viable population. And grizzly bears, of course, has been one of the big issues in terms of isolation of Yellowstone population. And Chris Servine and I wrote a paper 24 years ago now on genetics and conservation of grizzly bears. And so we've done some vortex modeling of grizzly bears. Just want to look at this really quickly. So these are 5. So Rich Harris did a lot of work when he was here as master's degree going out and trying to come up with vital rate estimates for Yellowstone grizzly bears so they could be modeled. So if we put those vital rates in with an annual growth rate of about 1.005, a lambda, this is the growth that you expect to see deterministically, and these are 5 different simulations. And you can see that generally the simulations sort of follow what you expect, but more importantly, there's big differences and between different simulations, and here's those 5 laid out. And the same demographic rates went into all 5 of these simulations. There's no genetics here. There's no inbreeding depression. But you can see what we see is demographic stochasticity. Just by chance, some populations get small, some populations stay large. The smaller populations are, the more likely they are to go down to 0 and become extinct. So demographic stochasticity is a problem, and genetics plays into that. So demography is the key. But if there is inbreeding depression reducing vital rates, then genetics can exacerbate that probability of extinction. So we have a lot of evidence now of inbreeding depression. So this is relative survival on the y-axis, inbreeding coefficient from 0 to 0.5. So this is the inbreeding coefficient you would expect between a brother and sister mating. And on the average, looking at 40 mammal populations, the offspring of full sibs have a 33% reduced survivorship from birth to 6 months of age. They probably also have reduced survivorship from conception to birth and from 6 months of age to maturity. But just looking at that interval, there's a 33% reduction. That's what we mean by inbreeding depression. And this was a paper that came out a couple of years ago. So based on this, lethal equivalence is just a measuring stick that geneticists use. And so the more lethal equivalence that a population has, the greater the inbreeding depression. So 33% reduction in survival from full sibs is what you would expect if there were 3 lethal equivalents. And looking at other parts of the life cycle, O'Grady et al. Concluded that on the average populations have about 12. So much, much more than originally estimated. And so here's a vortex simulation looking at 100 populations of grizzly bears, carrying capacity of 350 bears, using vital rates comparable for what we'd expect for Yellowstone. And we will have 3 different lines which represent 3 different amounts of inbreeding depression. So if we assume there's no inbreeding depression, Over 200 years, almost all the over 90% of the population still exist. So there's some demographic stochasticity, but most populations survive. However, if we put in a little bit of inbreeding depression, reduce the probability of survivorship of individuals who have related parents using LE lethal equivalents of 5, remember O'Grady says 12, so it's a fairly modest amount of inbreeding depression, we get a very different result. And there's two things, I think, that we see here. The first is that a little bit of inbreeding depression greatly reduces the probability of the expected probability of the population persisting over 200 years, so it goes from 90% to less than 10%. But the other thing is that we really don't see the effect until 100 years. And that's because grizzly bears live 25 years or so, and the generation interval is 10 years. So it takes a while for genetic inbreeding to build up. And so it's a longer-term process. So this is work we did a while ago, and it's been nice that this type of analysis has been taken into account in the conservation strategy. So the conservation strategy says 2 migrants per generation are needed to maintain the current level of divergence between NCDE and Yellowstone, and there are plans in place that if there's no natural gene flow to start moving bears around. But moving bears around can be really difficult, so I've been trying to push for artificial insemination for a long time, but and I hope I think it would be, seriously, if it were possible, it would be a lot easier than moving individuals around. So that's a nice story. Well, it's a story where the genetics, I think, made a difference, was taken into account by the managers and people that are writing conservation plans. A story which is not quite as satisfying from a conservation genetics perspective is West Slope cutthroat trout, native trout around here. Rainbow trout were introduced over 100 years ago. So western Montana and northern Idaho is West Slope cutthroat trout country. But there are regions where West Slope and rainbow trout both occur in eastern Washington and Oregon. But where they occur naturally, they don't hybridize. Or if they do, they hybridize, but it doesn't go very far. It's limited. They don't form these hybrid populations. But where they have not been historically sympatric and hatchery fish have been put in, I think everybody knows that hybridization has become common. So we started looking at this in 1973, going through all these techniques. Now Gordon Lukart is involved in trying to develop thousands of markers so that we can really understand what's going on in terms of hybridization. So this is work, just to give you an example of what I mean by hybridization and hybrid swarms. So this is a population over in the mussel shell. So using allozymes, looking at 8 different nuclear loci. A Y means a Yellowstone allele, W means West Slope. They also hybridize with Yellowstone, which unfortunately is confusing, but it's the same issue as with the rainbow. And if you look at all 15 individuals across here, you'll see that there aren't any Yellowstone cutthroat trout or there aren't any West Slope cutthroat trout. There's just admixed individuals. And that's what we mean by a hybrid swarm. Both of the original populations have been lost. We've created a new hybrid type. So we've been studying this since 1973, actually, before I came here. This is work done by Than Hitt, looking up in the North Fork of the Flathead, went out and sampled a number of populations, looked at, 42 populations, only 17 of which look like, using genetics, are still native West Slope. Most of them are hybridized, and many of the populations have become hybrid since 1985. So even though rainbow trout are no longer being introduced, the hybridization is still spreading at a fairly rapid rate in the North Fork, or at least it was. And this is work we did with Fish, Wildlife, and Parks. This is Brad Shepard put this together, which is a little bit daunting. So we've now analyzed over 20,000 individuals genetically, over 1,300 sample sites. And this number of fish per samples on the x-axis is this number of individuals. And you can see that these are what we consider to be unhybridized West Slope. About half the populations are now hybridized, but all these populations were sampled because people thought they were not hybridized, thought they were West Slope. So even those populations which are most likely to be West Slope, half of them or more are actually hybrids. So the biggest threat to West Slope is hybridization. And in 1997, Fish and Wildlife Service was petitioned to list West Slope. And they ruled 2 years later it was not warranted because of widespread distribution and current status. So they said, oh, there's West Slope trout all over the place. They were then sued because people said, yeah, but you're counting hybrids as West Slope when they're not. And so this went to court, and the judge said, yeah, you have a point. Fish and Wildlife Service did not use the best available science and ordered Fish and Wildlife Service to reconsider after taking into account hybridization. So Fish and Wildlife Service came to us and said, would you review the literature on hybridization, the effects of hybridization and the West Slope cutthroat trout? And we said yes, and we wrote this paper. And we got in a little bit of trouble because they just wanted us to review the literature and say, here are all the papers that dealt with the issue. But we felt that wasn't very satisfying, that interpreting the results was also part of being a scientist. So we interpreted the results, and we got in real trouble. Lynn Keating actually asked me to change our report because he didn't want that interpretation there, but we didn't change it. And we said, well, you know, there's three things you could do in terms of, so the issue was, what is a West Slope cutthroat trout in terms of the listing unit for the ESA? If it's only non-hybridized populations, then there's not many populations left in western Montana. However, if you include populations which have some admixture with rainbow or West Slope, or you include populations which just look like a West Slope, then there's going to be many more populations, and they're probably not threatened or endangered. So I think one of the problems here was determining the listing unit was going to influence the decision that they made, and I think that was unfortunate because I wish, from our perspective, I don't think listing West Slope is going to do any good. I think it's hopeless. I mean, you're not going to get rid of rainbow trout. So I'm not in favor of listing West Slope, but I am in favor of doing the science right. And I think scientifically, only West Slope cutthroat trout are West Slope cutthroat trout. I mean, I think that Ed Bangs, if there was a dog, if somebody brought a dog down here and it happened to look like a wolf, I don't think Ed would want to count it as part of the wolf DPS in the northern Rocky Mountains. So I think there's good reasons why we only should include West Slope as West Slope. And so that's what we recommended. And we said West Slope have been separated from other trout for a couple million years. They're evolutionary important lineage. And if we include the hybrid swarms, we're just going to be protecting those populations which are the source of the hybridization, which is a threat. So Fish and Wildlife Service didn't agree with this. And they said that we're going to include anything that looks like a West Slope morphologically. And this is really dumb. And I think first off, there are no morphological data. Nobody goes out and collects morphological data. As I showed you, we have 20,000 genotypes. There's lots of genetic data, no morphological data. Why would you use morphology? And if you do use morphology, we had shown that morphology can't determine whether they're hybrids or not. So I think this was not a very, I don't agree with their ruling. And they said that our recommendation was not valid. Well, okay, that's just what I said. They said our recommendation was not valid because there's no evidence that admixed populations are a source of spread, and there's no evidence of reduced fitness of the hybrids. So we just happened to have a number of studies online, and we first showed that, this is work done by Matt Boyer, who now works for Fish, Wildlife, and Parks, showed that indeed the hybrid populations are the source of hybrids. So that is a problem. And the second thing is Clint Mulfeld over at Montana State did a really nice paper, it was part of his dissertation, looking at the effects of hybridization in the wild, that it really does reduce fitness. So he went and collected all the fish in Langford Creek over a 5-year period. Microsats were done in our lab, identified the parents, identified the progeny, so that we knew how many offspring each individual had over a 5-year period. And this is the result. Mean number of offspring per female in the Y. X-axis is proportion of rainbow admixture. So this is a pure west slope. This is a pure rainbow. And you can see there's a huge decline in fitness as you have more and more rainbow. And Fish and Wildlife Service said, we'll accept anything with 20% rainbow trout as a west slope. There's a 50% reduction in fitness. And we saw the same thing for males, although not quite as dramatic, but still about a 50% reduction. So it's really frustrating to me that in this case, where I think the science is simple, hybridization is spreading because of hybrid swarms. Hybridization is reducing fitness, and I think it's really unfortunate that Fish and Wildlife Service made the ruling that they did. And I called up Don Campton, who is their primary geneticist and works for Fish and Wildlife Service. And Don and I are really good friends. We've known each other for 35 years, but we still disagree. And I asked Don, well, is it likely that, will Fish and Wildlife Service reconsider now, since you said you made that decision because we didn't have this evidence? Now we've published 2 papers that provide the evidence. And he said, no, we won't go back and revisit that unless somebody sues us, which I think is unfortunate. So I'd be happy to answer any questions. And as I said, one of the most satisfying things in my career is graduate students. So when I turned 60 a few years ago, Mike Schwartz, who had been threatened for years to write the book of Fred, so when poor unsuspecting grad students like Jody came along, they would know all of my idiosyncrasies. So he wrote the book of Fred, which is actually a valuable document, and I often refer my grad students to the book of Fred. So thanks. I'd be happy to answer any questions.
So if the hybrids have reduced fitness, what's keeping hybridization going?
Well, you know, those are my next slides, but I knew I couldn't put that in here. What's keeping hybridization going is that it's like we call it a genomic ratchet. So once you produce a hybrid, that hybrid can no longer, none of the offspring of that hybrid can ever be a west slope cutthroat trout again, because it's already hybridized and half of its genes are from rainbow. And so even if, and I can send you the papers on this, even if there's a 50% reduction in fitness, hybridization will still spread because all of the progeny of hybrids are hybrids. So it's not like Mendelian inheritance, it's sort of like, it's a mixture.
But if a hybrid then continues to breed with a pure, and their offspring grow with a pure, couldn't you theoretically get back to essentially pure?
There are 40,000 genes in the genome, and the hybrids, as that population I showed you in, I can't remember the name of the lake over in Musselshell, after a while there are no pure fish to backcross with. So we just have this mixture. And so you can imagine if you had 20,000 white balls in an urn and 20,000 black balls in an urn, how long it would take you to sort out and have only white balls come out twice. So 2 individuals mating are only going to produce west slope. So once you form a hybrid, you can't go back again.
A follow-on question from that, where the 2 species occur naturally together, Why is there less hybridization?
We don't know. We know they form F1s, and once in a while you see a backcross, but it doesn't go beyond that, and it's only a small proportion of the population. Rob Leary has lots of data showing that. There's another question back here.
Yes, it's related.
Okay.
I mean, have you looked at the fitness of the individuals where they're naturally occurring
No.
sympatric?
No. And it's, there's lots of things you could do. So up in the, I'm spacing out, what's the name of the river up in Libby? The Kootenai, thank you. Up in the Kootenai is where they naturally co-occur. So generally, streams are either west slope or rainbow, so that's one way, so they're geographically separated out. And there's also the possibility of different times of spawning. But we do know they do form hybrids, but there must be enough hybrid penalty with the low levels of hybridization that we don't see the hybrid swarms being formed.
Does it have the same effect with fitness reduction of crosses between rainbows and west slopes as Yellowstones and west slopes? Or is that kind of... I'd have to go, we have some data on that, but it's not coming, I can't recall. West slope, and it's another slide I took out. Yeah, west slope and Yellowstone are different subspecies, and subspecies often don't have much biological reality. But west slope and Yellowstone are actually as genetically different as rainbow and west slope are. So I think the same thing would be true, and it may even be, well, I don't know. You have to think of, all the Yellowstone came from Yellowstone Lake, where the west slope are living out in streams, and so I would guess the effect would be the same. Yeah?
So as a manager, what do we do? Because most of our west slope cutthroat populations are isolated, that's why they're still pure.
Yeah.
But now, usually it's because they're isolated behind some wooden crib dam that was put in 50 or 60 years ago and it's going to fall apart. Now we have to make the decision, are we going to open it back up or leave them isolated? But then of course, if they're isolated,
Yeah.
they have inbreeding depression issues.
Yeah, and that's a it's a double-edged sword. And you look at places where there isn't hybridization, there's places because there's a big dam like in the Hungry Horse. And I've been here since 1976, and Montana State Fish, Wildlife, and Parks has been great. I mean, they've been really concerned about hybridization, and I think they're doing as good a job as you could. So I have no complaints about management, and they're doing a really good job as far as I can tell. I guess my complaint was what I see as people making a decision whether or not to list, distorting the science to justify the decision that they wanted to make. And that's what really concerns me. Joel?
So you've given us like 40 years of change and the fascination with genetics, natural variability, kind of some Darwinism. Then you've taken it through management conservation. Where and how would you like to see genetics going over the next 10 years in terms of conservation? Just continuing this? Are there more?
Well, when they asked Jimmy Buffett where his music was going, he said south. But well, I guess so I'm teaching a grad class that Jody is in, a few other people here, and we're looking at genetics of harvested and exploited populations. And I guess the big issue that we've sort of focused on is looking at selective effects of fishing or hunting. We have a fairly good handle on the science. The difficulty is talking with people responsible for managing, and how can we do a better job of getting genetics engaged in actually the way things are managed. And we've talked a lot about why that's so difficult. Genetics is complicated. Geneticists don't communicate very well. It takes a long time for genetics to be a concern. So bighorn sheep get a disease, they're dead tomorrow. Bighorn sheep are inbreeding. It's going to be 75 years before there may be a problem, and the person responsible for managing them is going to be long gone. So I think there are good reasons why it's difficult to get genetics incorporate into management, but that's what I would like to see over the next 10 years.
You've actually watched it, though, obviously playing a bigger role. I mean, the two migration per generation, the grizzly bear, the West Slope. I mean, the examples you've given, but it's certainly there being used both legally as well as biologically.
Yeah. Yeah, and I mean, we've come a long way from N of 2 as a viable population.
So what do you see as the best roles of communication in genetics? Like what can you do to make genetics more prominent in management and achieve those roles?
I don't know, do you want to answer that, Jody?
I think it's the lines of communication often just aren't there between on-the-ground manager on the district and the genetics sort of seems to be partitioned more into the academic realms. A lot of the places we publish, like molecular ecology, which is not really on the radar screen of a lot of managers. So I think geneticists need to do a better job of tailoring publications to the appropriate audience so that the word and the information actually gets out versus sort of getting clogged in the sort of academic pipeline of this side of the world, I guess.
Yeah, and that really is, I asked Jody because that's the stuff that we've been starting to discuss and we tend to discuss the rest of the semester of trying to, how can we do a better job of exactly what you asked? Yeah.
One way to think about that too would be to go ask the managers. What problems do you need to solve? What are you concerned about for these particular species?
Yeah.
So instead of coming to the manager with, look
Yeah.
what we discovered, go ask the managers first what they need.
Yeah, and Fish, Wildlife, and Parks, in terms of fish, I mean, we've been working hand in hand for 34 years, and they came to us because we have this problem with hybridization, can you help us? And now Rob Leary is actually a Fish, Wildlife, and Parks employee, but he's based here in my lab. So that's exactly what we've done there. But yeah, that's a really good point.
Well, if that's all the questions. I'd like to thank Fred again for speaking this morning, and we're going to